Hello everyone, this is Gagandeep and this is a video log for Microbiology Project 2 by Gagandeep. The title of the study is to determine the effect of light and dark cycles on seed sprouting. Basically, this study was conducted to determine what are the effects of different types of light cycles on the sprouting of seeds. For this, various types of seeds were exposed to different lighting conditions which included complete darkness, complete light, and natural light cycles. The seeds used in this study were soaked in different soaking solutions which were water and 100 microgram per ml ascorbic acid solutions. The data collected was collected in the length of roots sprouted after 5 days. The data was collected using a simple ruler. These are the materials that were used in the study. For this particular fourth trial, we used mung beans, bengal grams, and lobia beans only. The soaking solutions used were water and ascorbic acid. Methods for soaking: 15 seeds each from each type were soaked in water and ascorbic acid for 24 hours. For plating, the styrofoam plates were used on which a uniform layer of cotton was spread to entrap the moisture. The seeds were placed over the cotton and then they were covered with a towel, so the water was sprayed on the towel so that the moisture can be retained for the seeds to sprout. This particular video is about trial 4. For the previous 3 trials, we have already uploaded the videos with results. This particular trial included 3 different types of seeds, which were lobia, grams, and mung beans. Plus, there were 2 different soaking solutions used, which were water and ascorbic acid. These particular results are for water. From this, it is clearly evident that dark conditions were much better for sprouting of all 3 types of seeds used in this particular trial over the light conditions. Also, when these results were compared to the ascorbic acid solution soaked seeds, it is clearly evident that the dark the dark conditions are still better than the light conditions, but the seeds soaked in ascorbic acid solution have a greater average root sprouting length than the water soaked ones. So, from both these results, it is clear that dark conditions are much better for sprouting of the seeds, as well as the ascorbic acid soaked seeds sprout much better and a higher rate than those of the seeds which were soaked in the water only. So from the previously present data, it is clear that ascorbic acid is a growth promoter, acts as a growth promoter for the seed sprouting. This effect can be due to the free radical scavenger or properties of the ascorbic acid. That means ascorbic acid, when the seeds are soaked in ascorbic acid, the free radicals in the solutions from the environment are scavenged by the ascorbic acid. Thus, the seeds are protected from the oxidative damage. Hence, these seeds sprout much better than the seeds which were soaked in simple water only. Plus, since the seeds under the dark conditions are sprouting much better, it is very clear that when we use the dark conditions, it is better to grow the seeds, which is almost equal, because before germination, actually before the photosynthetic process begins, the seeds are to be placed or planted inside the soil, which means dark conditions are obviously better. These are the references that were used for the reference to the methodology or the procedures followed. Thank you.